Okay, let's start. Hello everyone. Good morning. Welcome to join the Open Box Science Seminar. Today our speaker is Qin Yue. Yue is a bioinformatics and system biology PhD candidate from UCSD, supervised by Dr. Trey Liker. Her research interest lies in developing computational methods for building structural, descriptive, and functional predictive model of human cells. Her favorite gene is MGL, which contributes to the amazing smell of durian. Today, her presentation title is Towards an in silico cell. And Han-Yue, feel free to take control for your presentation.
Thanks, Zishan, for the introduction. Hello, good morning, everyone. Really happy to meet everyone here on Zoom and introduce our latest work, which if you're interested, please also check out the paper, which is a multi-scale map of cell structure fusing images and interactions. And just again, I am from UC San Diego Editor Lab, and so for today, like apart from like how we are able to like introducing you how we're able to use protein images and interactions to build a multi-scale integrated cell, I'm also gonna elaborate on like what kind of biological question we can ask with such a map, and also some fun, like I think pretty fun, like applications of such a multi-scale integrated map in other areas of systems biology, and also how we can we build this map leveraging the power of machine learning, and how we can return back to improve our machine learning with such a map. So with that, let's get started with some intro. So most of you probably know, we finally have a complete genome sequence for the human genome assembly, like a gapless, full-length chromosome sequence. But with that, although we know that what are roughly the protein coding genes that we have in our human cells, what we still just scratching the surface of knowing is how these proteins, how do they really come together to form the protein complexes, and how the protein complexes they assemble into the biological pathways, and how these come into finally at organelle level and eventually forming cell. And also not to mention these different biological structures or protein organizations, they naturally occur at different scales or resolutions inside the cell. And here I'm just illustrating some dummy skills for illustration. And in the past, biologists have used various technologies to help us learn more about the cellular structure that naturally arise at different scales and resolutions, and two of which are very popular methods are protein imaging and protein-protein interaction. So for protein-protein interaction, what you often see is probably this messy hairball view on the lower left here, where you often see each node representing a protein, and the edges between them represent whether there's an interaction or not. And this kind of network, it provides you a kind of local information of the protein assembly inside the cell, because if you think about this kind of physical interaction, they often occur at several nanometers. On the other hand, protein imaging, it gives you a much more global view of this protein's localization inside the cell, because each cell in the image is the image resolution is often a few micrometers, covering the major cellular organizations, such as the one I'm showing you here on the lower right corner. So here is a very typical immunofluorescence protein imaging. And also it's one of the images that we use in our study. So here the green channel shows you the protein of interest, so it's target protein. And at the same time, in addition to staining your protein of interest, you often also stain several major landmarks. cellular landmarks, such as the nucleus, which is shown as blue here, or the cytoskeleton, which is shown as the red here. And so by comparing to the landmarks, you're able to know like what is the relative localization or distribution of this protein inside the cell. And despite their vastly different resolutions, their vastly different format, these two kinds of data, they actually share one common information, and that is it both kind of gives you a protein, a coordination of the protein inside the cell. This coordination is probably more obvious in the image inside, where you kind of assign, like you imagine assign a coordinate to the protein by comparing its relative localization to the cellular landmarks. For example, the nucleus and cytoskeleton shown here. However, the protein-protein interaction also gives you a coordination of the protein inside the cell, and this is a much more local view of the protein coordination. And it's like, actually you can think about it as like you're giving the protein coordination based on who the nearby interactor of that protein is. And so this kind of gives us like it thought like, once we have like coordination of the protein inside the cell, like imagining you're on the XY scale and you kind of like each dot is a protein and you have a coordinate of the protein, then like can you start to like connect the dots and measuring the distance between them? And once you have the distance between a pair of protein, then maybe we can do some clustering techniques to identify the cellular organizations or the protein structures that occur at various distances. And this is exactly the key insight that inspired our studies, that both modality, they actually inform you a kind of protein-protein distance inside the cell. So to pilot our approach, we collaborated with the Bioplex team and the Human Protein Atlas group to pilot and to test out the pipeline. So as these two databases, as you can already see, like the Bioplex is the database about protein-protein interaction, and Human Protein Atlas is a collection of a lot of protein immunofluorescence images. And also, just want to further emphasize, they each is the largest database within each modality separately. And also one nice thing about like these two databases is that even though they have assayed the proteins in different cellular backgrounds, there's actually one common cellular background that they both use, which is HEC293 human embryonic kidney cell. Which is also what we focus on in our study, so that we make sure that when we're comparing the two data sets and when we're integrating them, we don't need to worry at least of the noises that coming from different cellular backgrounds. And just a little more about Bioplex. Their technique of assaying protein-protein interaction is a technique called affinity purification mass spectrometry. And what it does is that your protein of interest here is called a bait protein. And by tagging a specific tag to this bait protein and then introduce this into the cell, you're able to pull down this bait protein by using a specific antibody against the tag. And by pulling down the bait protein, you at the same time pull down its interactors inside the cell. And so this is how we're able to construct a mini diagram or mini network of these bait proteins interactors that it interacts to these different other proteins inside the cell. And so that is how you're able to eventually form this massive hairball by pulling down a lot of bait proteins. That's what the that's what the Bioplex team did. And so our approach, it consists of 3 steps. The first step, can we find a kind of a coordinate of the protein inside the cell? And so how we do it is that for each data modality, we derive a protein embedding from that data modality. So each protein, after this step, will have 2 embeddings. One is derived from imaging, and one is derived from APMS. And this embedding idea is actually quite often used in biology, and it's actually most prevalent also in the single cell field. So if you've seen like those single cell papers, you probably see the Tisney or UMAP plots, and those are actually two-dimensional embeddings of the transcriptomic profile. So in those plots, you are putting cell into an XY space, and if two cells are nearby each other, then that suggests that their transcriptomic profiles are very similar. So here the idea is the same, that instead of putting like each protein into a two-dimensional space, we're putting into a 1024 dimension space. And the idea is that if the embedding should put proteins close to each other, if the proteins share, for example, a similar global distribution inside a cell or a similar interaction neighborhood. And this embedding technique is able to allow us to really enrich or strengthen or extract for the signals that are most useful for our study, and allow us to ignore the noises that's kind of inherent in the data. So for example, in the imaging, one kind of noise that you can think about is like the cell orientation, whether it's orientated left or right. We don't really care, but we want to know what is the relative distribution of our protein of interest against the cellular landmarks, for example. And in the interaction space, what we want to know is what is the local and more global interaction pattern of this protein in relative to other proteins. And so once we have embeddings for proteins, we then use a random forest regressor to translate the embedding of a pair of protein into a general measure of protein-protein distances. And once we have protein-protein distances for every pair of protein, we applied pan-resolution community detection to assemble a multi-scale integrated cell. And just a bit more about this pan-resolution community detection. Imagine you have a kind of a magical column, and this magical column is our algorithm. So your input into this column, like all the predicted pairwise protein distances. And I'll say, okay, only give me the protein clusters where every single member is closely associated with each other. So this is how you're able to get these small protein complexes where everyone is like strongly connected or strongly interacting with the other members. But as you gradually dial down the stringency, you begin to like include more like distant interactions or more transient interactions, and then you gradually enlarge the protein communities that you identify, and those enlarged ones also contain the previously identified smaller ones. So how this done algorithmically is illustrated here on the right. So so imagine you're going from bottom from up to top, and you start with a protein distance matrix or distance network, and I'll say, okay, only show me the edges that represent the closest distance between the proteins. And this is when you're able to identify, like when you discard all the edges that denote lower distances, you're able to identify these clusters of protein that are mutually connected by edges that denote a short distance between them. And these usually are small protein complexes or small protein assemblies, but as you gradually lower your stringency on like what edges to include, you're able to gradually include the edges that start to denote longer or larger and larger distances or weaker associations among proteins. And so here, in the middle, you see that the red edges start to appear, and on the upper here, you're starting to see the purple edges. And the protein communities thus gradually include other proteins that are more distantly associated into larger and larger protein assemblies containing the previously identified smaller ones. So by applying this computational strategy to all pairwise proteins that we have, this is how we assemble the version 1.0 of our multi-scale integrated cell music. So music here is also shown in the network view, but it's a very different network view from what I have been introducing you previously, and also from the one I'm illustrating here, like if you look at the protein-protein interaction networks. So here in the music map, each node represents a protein community instead of a single protein. And the size of the node denotes how many members does this protein community have, and the color of the node denotes like whether this protein community has been previously documented or is potentially novel, and we should like look into to see what are the novel biologies that we are able to get from it. And also the edges between the nodes, it now denotes a containment relationship between the systems. And so just as a toy example, like acetylploids and nucleus, they're contained by cell, and here we call it the child system of the cell. And also just let me further remind you that if you kind of imagine zooming into each node, actually behind each node is a protein-protein distance network, just as the one I'm showing you here on the left that I've introduced previously. And so because of that, just further remind that we have a predicted distance for a pair of proteins. Can we now like estimate the size of the protein system inside the cell? And this is exactly how we layered, or how we determine the hierarchical, the height layout of each system inside the music map, and is what is denoting here on the right as size ladder. Is what is the estimated size of this system by music. And so how well do we do in estimating this size? So here on the left scatter plot, I'm showing you a comparison between music prediction to literature collection. So each dot in the scatter plot denotes a protein system or subcellular component inside music. And the x-axis shows you what is a predicted size of the system inside the music by music. And on the y-axis is what is a literature, what is an actually experimentally measured distance or diameter of this system from the literature. And as you can see, music accurately sizes biological systems across a wide range of physical scales. And also on the right here, I'm showing you an illustration of a cryo-EM model of a pre-catalytic spliceosome, further demonstrating that music indeed it actually sizes biological systems as well as capturing their subunit organizations. For example, U1, U2, they are the subunit of this pre-catalytic spliceosome. So what kind of novel biology are we able to get from such a map here? And like we explored various stories inside the paper, and today I'm just going to introduce you to one of them, which identifies RPS3A as a novel RNA splicing regulator. So RPS3A, by name, you could probably tell it's a ribosomal subunit protein. However, in our map, it's closely associated with 3 other known splicing regulators. And so this makes us wonder, like, maybe RPS3 has some unknown function that we don't know yet. And it's probably related to splicing. So to test out our hypothesis, we collaborated with our colleague, Dr. Jinyo's group, and his student Maya to carry out an enhanced UV cross-linking precipitation assay, ECLIPSE. So just a bit more about ECLIPSE. So what ECLIPSE does is like, so inside the cell, you have RNA binding proteins denoted as RBP here, binding to different kinds of RNAs, and by UV cross-linking their interactions and then pulling down the RNA binding protein using specific antibody, you're then able to identify what are the RNA transcripts that this protein binds to, as well as where exactly this protein binds to on the RNA. And so this also enables us to like actually profile each protein based on what regions on the RNA that this protein binds to, and by categorizing the binding sites into various different regions on the RNA. And so as you can already see here, like RPS3A, its profile is very similar to those of canonical splicing regulators, and it's actually very different to those canonical ribosomal proteins. And so we also like further compare the profile of RPS3A with all the ECLIPSE that is publicly available. And we indeed observe that RPS3A robustly clusters with RNA binding proteins that are specifically enriched for splicing regulation. So that is one of like the biological, like an illustration of biological insight you're able to get from such a map. And what other stuff are we able to use with this kind of multi-scale map? So another study from Edgar Lab, led by Dr. Fan and Mark, is using such a multi-scale map to identify protein systems that are under mutational selection in cancer. So here, imagine like the previous music map I'm showing you is flipped down, like it's and here is like on the top are the genes, proteins, small protein complexes, and on the bottom is the cell. But this is again a multi-scale map. And so you by like kind of like now you're seeing like what at what level on this multi-scale map you observe a strong mutational selection in cancer. And the benefit of this is actually it elegantly addresses the long tail problem in cancer. So the famous long tail problem is that in cancer, there are a lot of like frequently mutated genes, but there are also a bunch of genes that even though they're really mutated, but they just like, it's just hard for them to make the statistical significance for biologists to study them. And that does not mean that those really mutated genes are not important in cancer. And so what this study explored is how we're able to identify the protein system or the subcellular organization that these passenger mutation jointly hits inside the cancer, so that even though if you look at each member of this protein in the system, it's maybe really mutated, but if you look at the protein system itself, it is significantly mutated among cancer patients, and also indeed carry biological significance, as they have demonstrated inside the paper. And what else are we able to do with such a multi-scale map? So as I've very briefly introduced in the beginning of this talk, we are also able to use it to improve on machine learning. So during like the music construction, we have indeed leveraged the power of machine learning or deep learning in pattern recognition to help us extract the biological signals. But the kind of deep learning that we have used in our study are all the typical black box deep learning, where you know, like, okay, what is the phenotype prediction, but you don't know what exactly the machine learning or the deep learning is really learning from the data. You can only infer from it. And this problem is okay for our study because we don't need we don't really need biological insights, although it's good to have, but it's very problematic in the clinical setting because you really need to know what is the machine learning or the neural network learning in order to know like if we can trust the patient decision that it just made. So another study, a very interesting study from Edgar Lab, is by embedding this kind of multi-scale structure inside the neural network architecture, you're able to get not only the phenotypic prediction of this from the genotypes, but also know like what are the protein organization or what are the subcellular systems that at least the neural network thinks is important for making this prediction. And because you know what the system, so we call this a visible neural network, because you can see what are the important systems for this genotype to phenotype prediction. So just a summary, I've shown you like how we're able to construct a multi-scale cell map by integrating data from vastly different format and modalities, namely protein physical association and protein imaging data. And From this multi-scale cell map, we're able to systematically explore novel subcellular components or review pleiotropic function of the proteins. And also, as I briefly introduced at the end, how we're able to identify cellular system under mutational selection in complex diseases such as cancer. And furthermore, we can also bring visibility to black box neural networks using such a multi-scale cell map. And with that, I'd like to thank my advisor, Trey, and also Emma, who co-supervised this work, and thank all my wonderful collaborators without whom this work is really just impossible. And thanks all of you for listening and your attention. Thank you.
Okay. Thanks for US very nice presentation. And then anyone have any questions, feel free to ask question or type at the chat box if it is not that convenient to ask in person. Okay, yeah, I have a question. First, maybe, could I see the protein-protein interaction? Actually, as far as I know, there is a database recently, not recently, many years, a string database. It contains a lot of the protein-protein network. So what's the difference yours with that database? Actually, the string database also provided the protein-protein interaction or physical link. So is there some common or difference between yours or theirs?
Yeah, great question. So there are not only string but also various multiple databases of different types of protein physical interaction. Like for example, there's also a human cell map that uses biotinylation to probe physical interactions between proteins or the proximity between proteins, and there are also other techniques. So here, instead of using those, our rationale is we want to focus, is that when you combine technologies from different platforms, you of course strengthen signal, but at the same time you introduce a lot of noises into your data. And so as a pilot study, we're hoping to focus more on what we're able to get, because we want to also explore the relationship between such a between the interaction and imaging, because it's just two type of data of very different format and resolution. So in this pilot study, we want to see what at least are the conclusion we're confidently making before we jump into the vast field of all the different kinds of data. But as I've just mentioned, there are also other types, for example, biotinylation data that also tells you the protein proximity, and of course this method, once developed, can be applied to other data types as well. And also another thing about string is that it is of course a powerful database, but also by integrating all these kinds of all these databases, all the other databases, all the other data sources, it becomes hard to really trace back where exactly the signal is really coming from. And so here we really want to know what is providing us the signal. And so we focused specifically on one technology that's generated coherently from one single platform.
Okay, thank you. So anyone have any other questions? Feel free to type in the chat box if it's not convenient. Oh, here is a question. Yeah, from Wang Yueling. It says, very cool talk. A simple question. How do you estimate distance by using random forest?
Very, very good question. And I have a supplementary slide for answering this question. And so just remind everyone that here, we're using random forest regressor to translate embedding from a pair of protein into a general measure of protein-protein distance. So what exactly is our label during this supervised task is actually a very interesting observation that actually the number of proteins in a subcellular component is highly correlated to its actual physical diameter measure from literature. And we observe this in gene ontology. And as you can see in this scatter plot, so each dot here is a cellular component inside gene ontology, and the y-axis, so the x-axis is the number of proteins annotated to this system, and the y-axis is its actual physical diameter that we collected from literature. And so this next correlation gives us a calibration function so that we can convert the annotation between, so we can convert a pair of protein into physical distance by looking at where exactly is this protein annotated in gene ontology. And this is done more specifically by looking at what is the smallest size of the system that these proteins, that a pair of protein are co-annotated to. And using this size, we're able to then convert to a rough estimate or general estimate of protein-protein distance between them. And just to further just emphasize more that this is the protein system used here is an independent set from the validation set that I have previously shown you in the previous slides.
Okay. Thank you. From Wang Yueling, thank you. Thanks for explain. Okay, did anyone have other question? A question from from Jimmy. Oh, his name. His question is that have you considered introducing other modalities such as iron localization, or are the method not high throughout enough? If it would be possible, do you think we could learn more about gene regulation based on the protein communities they localize to? Hey guys, he's thinking of something like ironfish, but maybe those experiments are difficult to perform as a skill.
Another really wonderful question. Yes, I definitely think this method could be applied to other data modalities. And actually this method, like even though we are demonstrating it using imaging and APMS, it's actually flexible to different data types, and also like the number of data types, the number of data that you integrate. And in theory, it can extend to any data and any number of data to integrate, but here we are demonstrating hopefully the idea using two data of very different format to show the point. And RNA localization, I actually think is a very interesting data type to integrate, especially it definitely, I would say, provide a different aspect that's missing by the protein. For example, like I know relatively little about RNA localization, but I think, for example, in the neuron field, like the RNA is localized to where the signal is going to be transducted. So this kind of information, I do think is a very good piece that is complementary to the protein imaging and also to the APMS. I guess the real question then is like, how can we best embed the RNA localization data from the RNA fish experiment to represent this kind of biological signal. Another thing I didn't mention is that for each data type, the selection of the embedding, we compared different type of embedding techniques to select the embedding that at least for our use case best enriches for the biological signal useful for us. And so I think it's definitely interesting. I actually think it's high throughput enough. I'm seeing some like large databases of RNA localization, and hopefully like there are more coming up, and so we can really start to integrate and see where the signal of the RNA really is inside this kind of map. And I do think that it can give us more of our gene regulation information inside the map as well.
Okay, awesome, thank you. Okay, thanks for your nice explanation. And then if anyone have any question, feel free to just unmute and ask directly. Okay. Maybe I have a last question. Maybe. I just saw your one of the slides that validates your using the eclipse data from Eugene.
Yeah.
Yeah. Actually, the Eugene method they are using. Actually, the eclipse data they are predict the RNA and protein interaction. Here you are the protein interaction. So is there some how you validate this? Actually here I'm not very clear here.
Yeah, so here, like from our cell map, we identify like RPS3A closely associating. So actually these four proteins, they form a small protein assembly inside our map. But it kind of is like, then it's like RPS3A is the ribosomal subunit protein. Like if it's strongly associated with RNA splicing regulators, which are the three other proteins here, then it probably suggests us that it has also like RNA splicing regulator role. And so that is why to explore this function, we did a experiment to investigate the interaction between RPS3A and other RNA species to see if our hypothesis about its splicing regulator's role is making sense. So even though like we're looking at protein-protein interaction, this is an orthogonal validation of our finding.
Okay. Thank you. Okay. Thanks everyone. Thanks everyone for participating in today's seminar. And welcome everyone to participate our next Open Box Science seminars, and feel free to participate the Slack panel or the Facebook or Twitter or official website. Our next seminar will be listed there. And thanks everyone, and have a good day. Thanks you.
Thank you. Thanks for organizing.